Thanks everyone for being here for our first seminar of the spring quarter. We're really excited to have Professor Elizabeth Jerison with us, who's going to tell us about inflammatory response in the dynamics. Thank you.
Thank you. So thank you all for the invitation to be with you here today. So what I'm going to do today is three things, essentially. First, tell you a little bit about some of the general questions that my group is interested in, and then talk about two vignettes addressing some aspects of these questions, really trying to start to probe some of the internal dynamics of inflammatory responses. So our immune systems are able to do this spectacular thing where you get some infection somewhere, and this system is able to react quickly to this unexpected challenge without overreacting, usually. And so if you think about this, so how does the system accomplish this? So there is one extremely straightforward way that such a system could behave in principle. So for example, so in that sense, it could be entirely externally driven. And so what I mean by that is that there could be a set of, for example, molecules that are associated with a pathogen, something that's not supposed to be in your body. These are recognized by receptors on immune cells when this, thank you. So when the idea is then that when these molecules are present, then maybe some of these, interesting, I can't see it on the screen, but okay, maybe some of these immune cells are activated, they go clear the pathogen, and then once these molecules are no longer present, the response gets shut off. And so this seems like a system that could work to have you able to react to something that something unexpected, but without running amok. And what's so interesting about this system is actually that this is very far from the whole story. So to begin with, such molecules exist. This is an important part of the picture. So there are so-called pathogen-associated molecular patterns that are recognized by things called pattern recognition receptors on a whole host of cells, including a bunch of innate immune cells. These are quite important to setting off inflammatory responses. But as mentioned, this is the beginning and very much not the end. So to start with, there's another set of molecules that are called damage-associated molecular patterns, and these are endogenous molecules that are produced by the system itself. But beyond these DAMPs and PAMPs, there's a whole cascade of intracellular signaling interactions that results from this initial recognition event. And so in particular, for an inflammatory response, as mentioned before, initially there are cells, including so-called sentinel immune cells, such as tissue-resident macrophages here, that recognize a library of these molecules associated with either an infection or tissue damage. And these start now producing a bunch of signaling factors, including most dominantly cytokines. Some of the important ones are listed here, which then go and are received by other cells, which themselves start signaling. And one of the most important events here is that the endothelial cells that line blood vessels will open up gaps, allowing immune cells and become sticky, actually allowing immune cells to escape into tissue, as well as there's also dilation of blood vessels and fluid release out into tissue. And this is important both for clearing infections, but also these immune cells running around are actually quite damaging to tissue. Sorry.
Are we born with these
Good
mechanisms?
question. Yes. So by the time you emerge from the womb, you have absolutely all of this machinery in place. And so what I'm talking about here is the so-called innate wing of your arm of your immune system, and as opposed to what is typically called the adaptive part of your immune system. And the main distinction there is that these cells are born with a fixed set of receptors that are germline encoded. So these pattern recognition receptors, so-called, are fixed throughout your lifetime. As opposed to exactly. So as opposed to the adaptive immune system learns through past exposures by updating its set of receptors, essentially, in a host of beautiful mechanisms that I'm not going to talk at all about. So one of the reasons that this system is really interesting is that there are a number of feed forward interactions that are likely quite important to ramping up responses quickly. So one of the known types of these feed forward loops is listed here, that involves these two very important cytokines and their action on a transcription factor. But because of these internal nonlinearities in the system, I think it's fair to say that these inflammatory responses have a life of their own. And perhaps the cleanest experimental demonstration of this is that actually one can set off something that looks like a systemic inflammatory response without even introducing one of these pathogen-associated molecular patterns, put it merely by injecting the cytokine itself into the blood. So it's not externally driven in the sense that the secondary mediators themselves can set off this nonlinear response. The external molecules is unnecessary. Additionally, sort of quite interesting, I think, sort of circumstantially, is the fact that people over their lifetime who have various types of autoinflammatory disorders and autoimmune disorders will experience sort of random flares at various points. So what my group is really interested in is trying to probe the internal dynamics of this system. And one of the reasons that this is challenging is that this really crucially involves interactions amongst a lot of different cell types in situ. And so we'd like to be able to preserve these interactions because they're really, I think, crucial to this, how the system operates, and particularly to the sort of importance of these dynamics. And the other thing that we would like is to be able to start to try to introduce types of controlled stimulation. And for this, I take sort of inspiration from nonlinear dynamical systems that we understand quite well. So in the top here is a picture of a diode laser, and on next to it is an actual measured bifurcation diagram for such an optically pumped solid state diode laser. So this, for any given device here, so this picture is fairly complicated, right? So as a function of the frequency of light that you're dumping in and the power, you can get oscillatory behavior, you can get chaotic behavior, there's period doublings and various things that occur. But this is something that we understand well enough how to model and measure that you can add these here in this case, these bold lines are from a model, and the points are from a measurement on an actual device. And that means that you can, if you want the system to behave in a particular way, you know exactly how to dial in the inputs. But to be able to ask even about questions about whether or not there are abrupt transitions in behavior, as one might sort of fairly generically expect for such a nonlinear system, we have to be able to do something that's a little bit like dialing variables along this axis. So something like controlled stimulation. And so how might we make that a little bit more accessible? So I said, so this top example is a diode laser. I think actually really cool. This bottom example is a giant squid axon, a neuron, and that is an actual measured bifurcation diagram for a giant squid axon, which I think is pretty amazing that one can do that. Yeah. Yes.
The power and wave frequency are things that you control. So then what are the dots correspond to?
Yeah, so okay, so this is, so certainly very much not my experiment. So, but what's happening in what they did in this case is that they made a lot of observations of the amplitude output of the laser at different points that you're not seeing at different parameter values, and then they looked at when it switched, and so the points are actually the points that they're plotting here are intended to mark where the bifurcations are. They're when the system switches behavior. So a point here means that, so let's see. So this is this purple here is actually when the thing goes from being off to being on. For example, as you transition across one of these solid lines, that's how to read the diagram. So it's like, you know, if you're sitting in the middle of one of those regions, there's a particular behavior that is essentially true for that whole region. Okay, so, right, so the reason, and this is, I would say, just to mention before that, you know, so I think we're quite far from having this kind of a description for the immune system. This is, as I mentioned, more just inspiration for what I would be able to one day do rather than something that I can show you right now concretely for our system. So the main experimental model that we use to try to start to make accessible some of these controlled perturbations, as well as some quantitative measurements at larger scales, is the larval zebrafish. So the reason for the fish is that somewhat remarkably, they actually have all the same immune cells as mammals. Even the sort of adaptive immune system is actually quite well conserved. And the innate immune system is thought to be sort of even better conserved. And I'd be happy to talk more about that in questions if people are interested in a bit more about the biology. But so, and they have some real advantages. So one of them is simply size. So that's that little thing there, right, is an actual larval zebrafish to scale next to a penny. Think of them as like fruit flies, for example. And so that allows us to both do scales of experiments, eventually do large scales of experiments over many fish, and image things like large parts of the organism. And so they're also, optically transparent when they are young. And so this allows for doing a bunch of live microscopy, and also has the potential to be able to introduce perturbations with light, which is not something that, we have a bunch of work on that I'm not really going to be talking about today. But this is part of our motivation for why we use this system. And again, another is simply that depending on the types of things that you want to measure, you may want to be able to do scales of experiments that you just simply could not do with mice, because it would require, you know, walls and walls and walls of mice. Let's see. So what I am going to do today is to tell two vignettes about how we've started to approach a couple of the problems related to trying to understand the internal dynamics of the system. And the first one is to really try to ask a simple question, which is to start to disentangle the influence of the tissue and the cells within it on spatial distributions of gene expression response during inflammation from something complicated like the spatiotemporal dynamics of a pathogen itself. And then the second thing that I'll do is talk about some recent work of Nico's, who's here, to start to build some tools to essentially to be able to identify if you have, to be able to look at some data, which in this case will be sets of noisy trajectories, and identify what phase or what behavior those trajectories belong to. So sort of what zone are you in situations where we might not have very much idea about what the right model to write down is. So there have been a couple of questions. I want to also encourage you to interrupt me with questions as we go. All right, so the first story. So as mentioned, I was interested in this question of you know, if you introduce a pathogen, it has its own whole complicated spatiotemporal dynamics, which of course can drive spatial patterning in the system. And I was wondering how one might ask whether or not there are any inherent length scales associated with the response of the tissue on its own. And so to address this question, we're going to look at a very simple model system and a model inflammatory response. So our system will be the tail fin of the larval zebrafish. This will be inflamed because we're going to expose the entire fish to a uniform bath of one of these pathogen-associated molecular patterns. In this case, one called lipopolysaccharide, or LPS, which is sort of the most canonical pathogen-associated molecular pattern. It's a molecule that covers the surface of gram-negative bacteria. And again, we want to know whether or not it's safe to treat these cells as sort of independently responding, or whether or not there might be more effects due to the fact that they are part of a tissue in an organism. So as mentioned, our model is going to be bath immersion of these fish in lipopolysaccharide. And this is known to cause systemic inflammation, especially in this tail fin tissue. So to address what types of how these cells are responding, we're going to ask what genes they turn on in response to this LPS. And so to identify a good set of genes to label, we did a single molecule, a single cell RNA sequencing screen on dissociated cells, and chose essentially the top several genes that were overexpressed in the LPS exposure versus control. This actually turns out to be a fairly interesting list of genes. So it includes IL-1B, which is a cytokine that you saw on the previous slide, which is one of the most important pro-inflammatory cytokines. A couple of what are called matrix metalloproteases, which are enzymes that are actually released by cells and can go and digest extracellular matrix to give allow immune cells to infiltrate more easily. Something called PTGS2B, which is known as COX-2 in humans, which is the rate-limiting enzyme for the synthesis of something called prostaglandin. And this enzyme is actually the target of ibuprofen and other NSAIDs, as well as some other things that are associated with inflammation. And we added to this panel one inflammatory repressor, SOX3A, and then also just a gene that labels macrophages. So what we're going to do here is to expose these fish to lipopolysaccharide at a couple of different concentrations, and then at a particular time point, the fish are sacrificed and tail fins are fixed and mounted on slides, and we label against these different RNA species to ask what the cells are expressing. So this is what one of these measurements looks like. So here I'm labeling these two matrix metalloproteases. This was a tail fin that was exposed to LPS for 10 hours at a fairly high concentration. So you can see, so these ones happen to be nuclear localized, which is why you're seeing the cells so distinctly. So these things here are cells. The reason that there are spots is that we are labeling against individual RNA molecules. So the way that the measurement works is that we just have one color per gene. We image over 3 rounds, 3 genes per round, and then cross-register the samples using a DAPI stain of the nuclei. And then there's an approximate cell segmentation here, just in 2 dimensions here. And then we can either count spots or measure intensities associated with each fluorescence channel, which is then assigned to a gene to get a measure of the amount of that gene being expressed.
So these are static images.
Yes.
Is there time dependence to this? Kind of
Great question. So yes, okay. So we only so we had so the measurements that I did here are at all of the concentrations at 10 hours, and then at the highest concentration also at 4 hours. So we have limited resolution from this measurement about the dynamics. And these measurements are necessarily so the fish is sacrificed, that's the end. we have a lot of interest in following the dynamics of these processes. And so, at the moment we have, newer tools in the lab where we have, for example, fluorescently, for example, some genes, when they turn on, drive fluorescence of a protein that you can then image live. The disadvantage of that is that you can only watch. There's a few technical things, but one of the main ones is that you can only watch a couple of things at once. Basically, you're limited typically in the number of fluorescent channels that you can realistically monitor at the same time. And so you can choose a couple of your favorite genes or things to measure at once and get full dynamics. For some things that's absolutely worthwhile, but that's sort of the trade-off.
Yeah, because I was picturing like if it were oscillating or way.
Yes, that's right.
Those things you wouldn't
that's right.
necessarily know. Or...
That's right. That's right.
Yeah,
Yeah. And so, we have other experiments that are very much designed sort of more exactly to look at these types of phenomena. Yeah. Okay, so all right, so so at a basic level, what happens when you expose these fish to these stimulants? So these markers do turn on. So they turn on actually, interestingly, fairly abruptly over a relatively narrow concentration range, there's what looks like sort of a heavy tail of a relatively small number of cells that are expressing things very highly, especially at the sort of more intermediate concentrations. So the first question that we asked is simply whether or not there's evidence for actual differences in expression levels from cell to cell beyond what one might expect from counting noise and segmentation errors. And this is because, the simplest possible null model of what cells do when they see a uniform signal is that they all turn on uniformly, right? And probably not very surprising given literature from cell culture and other types of experiments. There is indeed quite a bit of variability or heterogeneity in how these genes are expressed. One way to look at that is to compare a null model where we try to take into account these things like finite counting errors and potential cell segmentation errors, and compare the width of the distribution of counts across cells in the actual data to this null. It's always the case sort of across these genes and samples, except for perhaps the most sort of highly activated samples that there's quite a bit more variability in the data than this null distribution. So there is heterogeneity there, and the main question that we'd like to understand is how this is spatially structured. So first of all, is there any evidence for spatial structure, or are these cells just spatially independent from one another? So to begin to address that, we can just measure spatial autocorrelation. How likely are you to be expressing something at a certain level given your distance from another cell? So there is excess autocorrelation for all of our genes compared with a permutation on the cell locations. So there is so the cells are not spatially independent. There is some role of spatial structure. So to start to ask what is the role of spatial structure? So there are sort of two main ways in which there could be spatial patterns here. One of them is akin to in development. It's possible that there are some cells in some part of the fish that express some of these genes and cells in another part of the fish that express others. So there's maybe anatomical correlations between these expression patterns. Or on the other hand, it's possible that there's spontaneous generation of correlated zones due to the biophysics of the tissue or interactions amongst the cells, or due to fields of secondary signaling molecules within the system. So to start to disentangle these effects, we'd like to ask whether or not there are spatial patterns that line up with anatomy. And so here we sought to map these tail samples to a uniform set of coordinates. And so in our system, the most important coordinates are how far you are from the edge of the tail, and then essentially how far around you are, so an azimuthal coordinate. So we used something called a Kuntz mapping to map these tail samples to rectangles. And then we can look at these expression patterns across each of the rectangles. So here what you're looking at is each row is a sample, and just to emphasize it, so we labeled these genes, these 8 markers together in every sample. So each row is the same sample, and then each column is a gene. And so I'd like to draw your attention to, and they're just ordered they're ordered by overall activation level. And so I'd like to draw your attention to a few features of this data. So the first is that it's not the case that cells have to be in a particular location in which for them to be able to turn on a certain gene. So we can see that because in the most activated samples, there are cells basically anywhere in the sample that become active. So it's not the case that there are particular anatomical zones that exclusively turn on certain genes. But on the other hand, there are anatomical biases. And one of the ways to see that is simply to look at the average pattern across all the samples for each of the genes, and which is shown in the bottom row there, where I've normalized by the standard deviation within each sample so that we don't get swamped by the higher expressing samples here. And intriguingly, there's actually, so there is a spatial bias for each gene, and interestingly, it's actually different from gene to gene. So you can see there's actually a distinct spatial pattern for several of these genes. This is also true within one sample. You will see that it's not the case that there's a single sort of inflamed zone that turns on everything. Rather, there are actually somewhat distinct but overlapping zones for these different genes. So what we conclude from this is that anatomy biases, but does not sort of deterministically tell you whether or not a particular gene will turn on. So because anatomy doesn't fully explain these spatial patterns, we sought a more flexible tool to look at their structure. And in particular, we were interested in being able to ask the question of whether or not there's inherent length scales associated with this tissue response. And so to look at that question, we used some tools from spectral graph analysis. So this is something sometimes called graph signal processing, where so we'd like to do something that's like Fourier analysis on this type of sample, but they're small and they have funny boundaries and so forth. And so we do something that's closely related, which is that we simply draw. We represent each sample as a graph where each cell is a node and their neighbors are connected by edges. It's completely unweighted. And then you can write down the adjacency matrix. And in fact, we look at the scaled adjacency matrix here and ask for its eigenvectors, which are also the eigenvectors of the graph Laplacian, right? So, and this gives actually a really nice, so this is what you're seeing here is just a heat map representation of those eigenvectors on the samples. And this just really gives us a nice way to decompose these samples into different length scales that respects the geometry of the sample.
This isn't like PCA, it's more like point.
Yeah, that's right. Yes.
D like the degree matrix or something?
Yes, D is the degree matrix. Yes, sorry, I should. Yes. Yes. Right, so once you've done that, you could, so these eigenvectors are a basis onto which you can project your signals. And so one nice thing that you can do here is use this as to pull out sort of larger scale patterns if you're interested in. So this is the low pass filter where what we're seeing in the right hand column is simply the projection of the raw signals onto the first 50 modes. So there's the same number of vectors as cells in the sample. So typically, around 700 in our samples. And so in addition to that, so now we can ask questions about which length scales are responsible for contributing the most to the response. So to do so, we looked at the power associated or the coefficient associated with each of these modes. And so what you see here is this is sort of not very much not scale-free behavior. There's essentially a characteristic domain size where there's a relatively small number of longer modes that are contributing more to the signal in these different genes. And then there's a transition to uncorrelated fluctuations, where again, the gray come from a spatial permutation of cell locations in the samples. So from this, we can pull out a length scale associated with just the scale of that plateau size here. So I'm calling this the domain scale in this lower plot. And this is, so this ranges across the genes and samples, but it's on the order of about 100 microns, which is, roughly 10 cell lengths, although actually these epithelial cells are a bit bigger, so one could say 5 to 10 cell lengths. And finally, this gives us a fairly natural way to quantify how much these larger scale effects contribute to the variation in gene expression that we observe, as opposed to cell fluctuations. And this is because the fraction of the variance explained by the long modes is simply the same as the fraction of power summed up through a certain mode number, which is what you're looking at in the top here. And so from that, we see that in general, these longer modes explain most of the variation in gene expression. Although I would say that there is still quite a bit of contribution from cell-to-cell fluctuations. So and this tells us that interestingly, the tissue, there are these fairly characteristic length scales, and the tissue is a predominant factor in determining patterns of gene expression, even in the presence of this uniform stimulus. So a summary of what we've learned. So we learned that the tissue can induce spatial structure. There's a somewhat typical domain size. These account for a majority of the variance in gene expression. And interestingly, and I think surprisingly to me, there's overlapping but distinct domains for each gene. So it's not the case that, so these cells start expressing these inflammatory genes, but it's not the case that there's like one program where you turn on all of them together. In fact, there's actually variable expression from cell to cell. And this is due, so there are biases due to where you are in the tissue, but there is also sort of idiosyncratic appearance of these sort of little zones of inflammatory expression. And some questions that remain open. So why do you have these domains forming? So one possibility is that there are, which is actually the one that if I had to put money, I would favor something along these lines, is that some cells are primed essentially to start expressing certain genes due to, for example, their chromatin states. And the reason that there can be spatial correlations in this is because this is inherited over short terms. And so potentially during the development of the tissue, you actually have little zones that are more primed to make one thing versus another. Another possibility is that there are things like auto-amplifying interactions that where things interact spatially and encourage other cells around them to start making the same things. With a protein like IL-1B, this is potentially possible. And distinguishing these types of effects, dynamical measurements would help a lot to distinguish these types of effects. Another question is, so is this functionally significant? The real answer is that I don't know. One possible way in which this type of effect could be interesting is that it suggests that the tissue can perhaps auto-generate gradients of secreted factors due to these variable zones where these factors are produced. Okay, so I am now, I guess I have about 20 minutes left. And so actually I'm happy to pause and take another couple questions on this, or yeah.
The template itself, like,
Yes.
is it for sure uniform, that all the cells are really receiving the same? Isn't that like the geometry itself could be?
Yeah, so I have a couple of things to say about this. So yes, I agree. So certainly we cannot exclude do I want to go all the way back to the pictures? So certainly we can't exclude that there's a non-uniform distribution of LPS getting to the cells in the tissue. However, this on its own can't cause all of the spatial patterning that we see, primarily because there's a different pattern for each gene within the same sample, right? So if you had I think it would have been perfectly reasonable if you had a maldistribution of LPS, you could get one large scale spatial pattern where some areas are just more inflamed than others. But that's not what we see. And so, the multiple different large scale patterns, I think, means that those can't at least all be caused by a maldistribution of LPS, but that could be happening. And it is certainly not unlikely that there are differences. Yes.
Different patterns.
Oh, so yes, but then yes, you have to have that plus some interactions, like some secondary signaling in the system, right? That's all. I'm not saying that you could potentially, yes, you could have a model where difference of these factors reacts differently, and there are additional interactions amongst cells. But just if in the simplest case where you just have a signal and everybody's responding and there are no additional interactions among cells, I think you can't do that. Is that?
Yeah.
Maybe.
or yeah, among different, no interactions among different genes.
Yeah. Or
Okay, no,
yeah,
I mean, with you.
Yeah.
So speaking of interactions among different genes, like, have you looked at how correlated the different genes are to each other?
Yes, I have. And so, in fact, so the short answer is there are correlations, but it's messy. It's not. And so something that I tried, but I would say didn't get a clean answer from, was to see whether or not there are certain, maybe there were certain sort of linear combinations of genes that explain some of the large scale patterning, right? So, okay, maybe, for example, you could imagine, so maybe it's not just you turn on everything all at once, but maybe there's there's one secondary signaling factor that turns on X, Y, and Z, and one that turns on P, Q, and R. And if you overlay these patterns on each other, you get what we see, right? So that's sort of a simple version of. So I looked into this a little bit, and I would say that there's nothing super clear along those lines. But there are sometimes like particular areas which I suspect have secondary damage where like many of the things are on at once, for example. So I suspect there's multiple of these things going on, but there's not there's not a super clear like, oh, these three genes are actually, strongly correlated module and, or these are exclusive to one another or anything like that. Did that address your
Yeah.
question?
Sorry,
Sorry, do you have a?
I
just want
don't,
to
yeah,
take
no,
it.
I did it and then I did.
Okay,
Yeah.
so you can ask me after, but.
This innate
Anthropology knowledge, and I was just wondering, there's sort of like more, maybe a leaf is a more uniform tissue, like similar experiments have been done in plants, maybe coarser readouts, just like cell death, whether or not that distribution is.
I have no idea. It would be interesting. I love plants. It would be interesting, but I don't.
Like the
I don't know.
terminology is very similar.
Yeah, that would be cool. That would be cool. All right, so I want to tell you at least for a few minutes, and you're welcome to ask me questions after or ask Nico questions about some work that we've been doing to address the problem of in this particular context of trajectory data, which you'll see we'll be looking at trajectories that live in the plane, of how do you identify which phase you're in, or which part of the bifurcation diagram. I'm using the word phase a little bit loosely. Okay, so for this talk, I'm going to situate this with an immune example, although the problem is more general. So suppose you had, for example, 3 groups of people, and you drew blood from them and you measured levels of 2 different cytokines, A and B. So you might see something a little bit like this. So in your first group of people, now where each of those orange curves is meant to represent one person. And you can look at these and scratch your head and say, okay, they look, there's some interesting dynamics. They look kind of different from one another, but of course we'd like to do a little better than that. And so we'd like to characterize what behaviors are going on here and relate them to one another. So probably the first thing that all of us would think of doing is to try to fit some kind of a model. And of course, so one could explicitly try to fit an ODE model, for example, or maybe an SDE model, because you can see that they're a bit noisy. And so this can be challenging if you're not sure exactly what model a priori, what model to fit. And in addition to that, fitting such a model doesn't actually answer the question of, is this an oscillator, or how do I put a label on qualitatively the behavior? And that actually on its own can be challenging if there's a very large number of parameters in the model. So a completely orthogonal approach to such a problem is that you could just nowadays throw these trajectories into a computer and ask for some kind of dimensional reduction or do some kind of machine learning. And people have done have tried such things with, for example, animal behavior and some trajectories of immune cells migrating to try to segregate different behaviors. And so what we're going to do here is essentially try to bridge between these 2 different perspectives. So our goal is to take a set of trajectories and be able to say from that, give a label to the behavior, like for example, is it an oscillator, is it bistable, etc. So to use machine learning, but in a way that sort of preserves enough of the flavor of some traditional dynamical systems analysis that we can maybe learn a little bit more about how these things are related to one another. So to start doing this, first we need a library of different dynamical behaviors. So again, the problem that we're going to be looking at here is simply dynamics in the plane. So there are 2 variables. And so to generate a wide zoo of different behaviors, we are going to integrate these SDs that are up to, so that there are polynomials with up to 3rd order nonlinearities, and they're restricted to be bounded. And so so this generates a sort of a wide range of behaviors, which one can again squint at these and say, they do a bunch of different interesting things. Yes. Yes. So right. So the white circles are unstable fixed points, black circles are stable fixed points, and the color is time. And the trajectories are all initialized at a random fixed point within each system. So in each system, so each of these pictures here is a particular set of parameters, and the trajectories, and then with 30 trajectories.
You just sort of sampled over the possible.
Yeah, so right, so yeah, so for the yes, this is a great question for right now. So what we're going to do is, or what Nico did is to train a neural network with 10 to the 4 such systems, which have randomly sampled coefficients with Gaussians. So you know, some spread, but so yeah, so it's a random library of these polynomials. Okay, so Nico's insight was that what we mean, you know, that one of the lessons from something like bifurcation theory is that much of what we mean by behavior should be invariant to various types of transformations that you could do to the system. So, you know, the behavior is the same if you rotate it, and maybe if you do some even slightly more dramatic things, you would like those to be recognized as sort of the same thing. And so what we're going to do is to use contrastive learning to train a neural network to recognize that a set of trajectories and its augmentations are the same thing, and then anything else is different. So the way that this works is that each so the input is a set of in this case is 30 trajectories that were generated from these SDEs with particular fixed coefficients, and then each of these sets of trajectories is subject to linear augmentations as shown there, so they can be rotated or reflected or sheared a little bit. And then there's a loss function that asks for a set of trajectories and its augmentation to map to the same point in latent space, and anything else to be different. And so this then once trained, it generates something that's a function that takes this set of trajectories as pictured in the bottom here and outputs a point in this latent space, which in this case is 128 dimensional. And when we look at for visualization, just various dimensional reductions of this. So on the right here is a first sanity check just from essentially a fairly sort of canned example, which is different points, different parameter choices in the Fitzhugh Nagumo model that are known to produce different behaviors. And you can see that, you know, it's not doing something ridiculous. These points, in fact, lie in different parts of the latent space.
Your augmentation doesn't do anything with how you time parameterize the trajectories.
Correct. So they're all 100 time points, and oh yeah, there's also like a permutation on the order. So the order of the 30 can't matter. But otherwise, yes, there's no dilation in time at all.
So how do you decide what are the positive pairs here?
So the positive pairs are the initial set and an augmentation and randomly so again the essentially a randomly chosen augmentation of the same system. And so then so in each round of training there's a batch of I think it's 2000 in this case, but of the input systems, each of which is like a set of parameters. And yeah, it asks for then that one and its augmentation to be the same and everything else to be different.
So you mentioned that your latent space is like 100 and something dimensional, so you're really projecting up into a much larger space.
No, I mean, so the trajectories, right? There are 30 trajectories, each of which has 100 time points, right? So that's a lot more than 128. But it is, I mean, it is still a very high. Okay, so looking at some slightly more interesting examples, so we can take some models with first of all, a model with a known phase diagram. So this is this so-called Stick-Hopf model is a model of non-reciprocal dynamics, and on the left is you can calculate a mean field phase diagram for this And so it has because of the non-reciprocity, it has a limit cycle in some regions, and it has a monostable region and a bistable region. And so what Nico did was to simulate a grid of these parameters, or and these, again, these trajectories have some noise also, and then ask via k-means clustering for in this case with k equals 3 for these different behaviors, and then you can fill in in some sense an automated phase diagram on the right. And so in this case the neural network does indeed recover the correct phases here. And I'll note there's some encroachment of some zones on each other. So in particular, you start to see oscillations a little bit before you get to the hop bifurcation that separates the monostable from the oscillatory region. So this is maybe consistent with something like noise-induced oscillations really before you get to the hop bifurcation. And similarly, you can see the monostable region encroaches just a little bit onto the bistable region because you can't quite resolve just after you pass the pitchfork bifurcation. You can't quite resolve that you have two minima yet for just a little bit of extra time because of the noise in the system. Yeah.
Some difficulty at the boundaries just because you have a finite time trajectory, and if you're approaching bifurcation, you might have like slow.
Yeah, that's yes, absolutely. And so there's always, I mean, for sure here there's you know, if you didn't see it, you didn't see it, right? So and yeah, that's just a limitation I think overall. Yeah. So it's just so it's written, it means just white Gaussian, so it's just a randomly sampled Gaussian variable. Okay. Yeah, so another slightly and so I just another thing that I want to emphasize is that throughout this, actually the network was trained once on the random library of polynomials, and then we're just using it. We didn't retrain it on these different examples. So another example here is looking at two variables from actually a 6-dimensional cell cycle model. And this one falls outside of the class of polynomials on which the network was trained. And so this is by some analysis, right, in the original paper that proposed this is also known to you know, so he calculated and thought that there should be two monostable regions and an oscillatory region, and he calculated these dashed lines as the boundaries. And he also thought that there should be an excitable region just past one of these boundaries here, and that is what is recovered again by this neural net, which picks up this excitable stripe here, and then these other regions. So the fact that we're seeing these different sort of phases that are segregating suggests that even though the neural network was not told about anything about the topology to begin with, it potentially learned something about the topology. So to take a closer look at this, one thing that we can do is to go back to our library of random polynomials and now color the points based on their fixed point structure. So what you're looking at in the left hand here is, so this is again one of these dimensional reductions, but where now we're coloring points based on the number of stable and unstable fixed points in the system, focusing only on the simpler systems that have up to 2 fixed points of each type. And they come in their odd numbers here because of the boundedness, the requirements of the system. So you can see that there is some segregation based on the fixed point structure, and indeed, and actually, you know, there are regions that seem to perhaps correspond to or sort of boundaries that maybe correspond to different types of bifurcation. So, for example, the the boundary between purple and green over there is where you would find a Hopf bifurcation going from monostable to oscillatory. But we'd like to be able to look at this not sort of in a projection, but in back in the real latent space. And one way of doing this is that we can, for example, go on a little walk-in parameter space in the SNCC Hopf model. So this is what's shown in the next panel here. As we travel, we're traveling in parameter space along this semicircle that's pictured up in the right corner on top of the mean field phase diagram. And you can see that, and then at each point calculating the cosine similarity, so essentially the distance between that and the next point in the latent space. We see that as one might expect, that when you go through where a bifurcation is supposed to be, you get a jump in behavior. So the latent space variable jumps. And this is, although this is much less true for a continuous bifurcation, like the one at pi over 2 there. And so the other thing, so one can ask, okay, can you use this as essentially a classifier to tell you what your fixed point structure is. And so this works decently well. So one can ask for a set of polynomials that were not included in the original training data. How well can the system automatically detect the fixed point structure? And of course, the ability to do this degrades with noise, and there are some more confusion between some classes than others. And part of this is because the fixed point structure is not a full determinant of the behavior. But we can get at least sort of a decent guess about what is the underlying fixed point structure of the dynamical system. So the final thing is that sort of interestingly, I think a strength of the contrastive approach is that it's able to sometimes pick up fairly subtle differences in dynamical behavior without having to design a specific test. So a cute example of this, so we can look at two systems that are the same except for the red terms in the equations there. So one of these systems is simply linear. And the other one has spikes, but we're looking at it before it has spiked. So we're looking at it at a period of time where all that happened was it was just each of these systems was exploring its own basin. So you can look at the trajectories and say that you can tell visually that they're somewhat statistically different. So the nonlinear one is feeling the nonlinearity a little bit. But it's not totally trivial to tell these things apart and to say that they're different without designing a specific statistical test for this, which you could do. This would, if you knew what you were looking for, you could design something based on like higher moments of these distributions or something like that. But so a nice thing is that you can just put this into the neural network, and it does actually pull these different things apart in the latent space. Yes.
So the picture on the left with the threshold, you're saying that's for your system with the red term.
That's right. Yeah.
And what you're saying is like, even if I just look at behavior near the fixed point, just the linear problem versus the one with the red terms, I can tell it.
Yes, that's exactly right. Yes, that's exactly right. So, so in conclusion of this part, this contrastive cartography is, I think, a nice tool for classifying these sets of stochastic trajectories without prior model knowledge. It gives you a location in latent space that characterizes the behavioral state, and gives you perhaps at least a decent guess as to the topology of an underlying two-dimensional dynamical system. Something that I didn't talk a lot about is that one can use this, so one can put now a sort of a system with an unknown where the models are not known into this neural network that was trained on the large library of random polynomials and pick out a polynomial system that has very similar behavior, if that's something you're interested in. So, you know, maybe a simple phenomenological or very sort of mathematically tractable model, but that at least reproduces some of the behaviors that you observe. And one could then again use that to you know, for example, design further experiments to see whether that was actually a good model for the system. And then another thing that I think is nice about this is that it is able to distinguish fairly fine variability between behaviors without having to design a new statistical test for each new behavior of interest. With that, I'll thank, so a bunch of the experiments that I talked about in the first half, so some of them I did as a postdoc at Stanford. So thank my advisor there, Steve Quake, in that lab. And then the latter part of the work has all been done, and I should have mentioned more clearly earlier, there's pictures, but in collaboration with, well, so Nico is the hero of this project. He's sitting right here. And then also with Aaron Dinner and his graduate student, Chris Chi. And then also a number of other people for lots of good discussions, et cetera. Thanks. I guess we're already over time, but please feel free to email me if you have additional questions.